So welcome to CS372. And today we are very glad to be able to invite Professor Melissa Gao to come back to give us a lecture on cancer biology again. I met Melissa about 2 years ago. I took her course at the Stanford continuous study session. And then last year we invited Melissa to give us 2 lectures, and I learned a lot from her. And she graduated from MIT in major in biology, then finished her cancer biology PhD program at Stanford. And she's right now a fellow of science and engineering education. So Melissa, please just take over. Thank you so much.
Yeah, thank you for having me again. Welcome everyone. Good morning. Hope you're doing well. So this is the first of a 2-part little series that I'll be doing this Thursday and then also next Thursday, just really thinking about cancer. I will give an introduction to a lot of aspects of cancer that are important. So if you don't have a lot of biology background, I'll try to provide the context that you need. But we will look at sort of the front end of cancer, which is how it's caused, how it's diagnosed. And then next Thursday, we'll focus a lot more on how do we then treat it based on all of this stuff that we've learned from it. So the agenda for today is I do want to give you sort of the background that you'll need. So thinking about how cancer is caused, we'll talk about things like carcinogens and mutagens. We'll talk about the genetics of cancer. So thinking about how mutations in genes contribute to cancer, which is how it contributes to cells acting really abnormally and becoming what we think of as cancerous and all those sort of traits associated with cancer cells. So we'll connect the genes themselves to what cancer actually does and how it acts. And then we'll start getting into, like, with all of that sort of foundation, we'll start getting into the more data-driven side. So we'll talk about the different ways that we characterize cancer through data and thinking about how we diagnose cancer and how those two relate. So how do we use lots of different big data and big data methods to maybe diagnose cancer better, either more accurately, earlier, or as we'll get to next week, like diagnose it with an eye towards treating it better. So that's really the goal for today. And then, as I said, next week we'll focus much more on the treatment piece. And also next week, there's a lot of sort of open-endedness for people who have questions or they just want to ask about connecting, if you're more from a CS engineering side, like how do you connect to careers that relate to this kind of work? So just a little bit about me. Thanks for the introduction. So I'm just sharing a little bit more about myself. So I'm a local. I grew up in the California Bay Area. I did go away for college, but I came back for grad school and I've been here since. As mentioned, I'm a science and engineering education fellow in the bioengineering department. And what that really just means is that I work on education for that specific department. So I focus a lot on what is the goal of a bioengineering education? What do we want our undergrads to actually get out of doing a 4-year degree program? But my background was very traditional, sort of cancer biology. I did major in biology. Like, I work in education a lot now, but my background was all first wet lab biology and then computational biology. And so what my own research was about was trying to use methods like visualizations to understand why some cancer cells would die with treatment, but others would resist. And so these bottom pictures here are sort of hinting at that I did a lot of graph visualizations to try to figure out what was so special about these resistant cells. And I just want to say I am very happy, as always, to talk to students at Stanford. That's one of the joys of my work, is getting to interact with students. So if you want to chat at any time or connect, just feel free to email me. I've put my email here, and I'm sure that it's probably listed somewhere, or because you're Stanford, you could look me up and shoot me an email. So I'm very welcome to that. All right, let's get started. So let's talk a bit about cancer. So if you're not familiar with sort of the story around cancer, which I think is a very dominant thing in the US, and it tends to be the dominant thing, or it used to be the dominant thing in countries where, for example, infectious diseases or diseases of childhood are less of a thing, like in what we would consider, quote, quote, the developed world. That's where cancer tends to emerge as then the thing that preoccupies society. And that's largely because cancer is associated with people getting older. So in general, in countries where they don't worry as much about diseases that affect youth and where people have longer life expectancies, that's in turn where you tend to see more and more cancer. And at some point, you start to feel like you know lots of people who have at some point fought cancer, or at some point died from cancer. And so currently, I think the statistics are something like 1 in 2 or 1 in 3 Americans in their lifespan will get cancer. So it's a very large number. And as you can see, it's age associated. So something is happening as we get older. It's true that there are some young people who get cancer, like children even, but by and large, we're looking towards like middle age and sort of into 50s, 60s, 70s is where you get cancer. And then it kind of falls off, I would say, probably in large part because those folks are less likely to get diagnosed with cancer. And even if they're diagnosed, there's sort of all these other I believe comorbidities, like other conditions that they're dealing with. So it kind of falls off because they're less likely to catch the cancer and actually be trying to fight the cancer as opposed to dealing with other health issues. So let's talk a bit about what's happening as we age. And so first off, as we age, you're sort of accumulating your life experiences. So you can think of it as you're accumulating exposure to stuff. So as we live and navigate the world, we're going to be exposed to carcinogens. I know the joke in California is that we have the little proposition signs that say, "Warning, something in this building will cause cancer." That in some sense may seem overblown, but it's kind of just like a byproduct of living in the world where there's a lot of pollutants or there's a lot of chemicals that maybe we haven't even identified yet that might have some role in promoting cancer. And you just kind of run into them over the course of your lifetime. And it's sort of just incidental that here and there you might run into a chemical that then causes a mutation. So I just want to give an example. Actually, this isn't even a chemical. This is UV radiation. So if you just get a lot of sun exposure to the extent that maybe you're getting sunburned, that is evidence of, being exposed to something that has the potential to cause a mutation. And so what I'm showing you in this picture is just this idea that some of these chemicals and the way they interact with your body might be then interacting with DNA in some of your cells, right? So if you're getting UV radiation from a sunburn, chances are the radiation is acting with the cells on your skin. And so it could be causing mutation in your skin cells. And so what's happening, generally speaking, in a lot of adults as they get older and they navigate through life, they take a plane trip, maybe they smoke, depending on their lifestyle choices, they go to the beach, is that they're accumulating mutations over time. And it's the case that we'll talk about that it's not just 1 or 2 mutations, it's kind of a kaleidoscope of mutations kind of all coming together. And that's really what leads to cancer. So as we age, it's kind of like accumulating these little genetic tweaks and hits. And if we get enough of them in the right constellation, then cancer arises. And that becomes just more likely as you get older. So let's talk a bit about mutations. So we're exposed to these things, exposed to the sun, exposed to these chemicals, our DNA changes. So what? So we're going to connect that DNA change to what we actually get out with cancer. So if you're not super familiar with why DNA matters, you can think of it as DNA basically is the recipe book. It is the instructions to make almost anything that matters in the cell in terms of, the recipe to make things like proteins or things like RNA. And the protein and RNA is the material that does something, right? So when a cell is just sort of sitting there and it just has all of its DNA, if the cell needs to send signals, if the cell needs to grow in certain shapes or directions, or if it needs to divide, all of those things would happen, like that action would manifest as a result of the DNA providing the instructions to make proteins. So we call this the central dogma, which is the idea that you have your DNA, but that is used to make RNA, which is then used to make protein, and the protein does something, right? So pretty much anything you can think of, chances are a protein has been made to help facilitate that ability, right? So it could be something structural, or it could be something like behavioral. All of those come about through gene expression, which is what we call this pathway. And so the reason why mutations matter is because they change the recipe, right? So I'll just give you an example here. There's just a way that we know to read the code of what the mRNA says in terms of these letters. And that sort of has an output, like an expected output in terms of the recipe for the protein. And when we have a mutation, it changes the recipe. And so it could be the case that it's just a very small change, like basically switching out an ingredient. You could think of it as you're baking cookies, instead of using brown sugar, you use white sugar, but it could be something really catastrophic. So the way I told people is that maybe there's just a slight mutation, but it's kind of the same as if you were reading a recipe for cookies, and then the pages got stuck together. So you flip to the next page and then you started making croissants instead. So there's this real potential to sort of have these genes that are just completely disrupted. Like the recipe is almost nonsensical after a certain point. And as a result, that protein is just different. The way it functions could be very different. It's possible to have mutations that don't change the function of your proteins, but there's lots of mutations that could make the protein not work or work better, which sounds a little strange, but we'll talk about what that means when it is overactive. All of those, depending on what the gene actually normally does, could be a problem. Are there any questions up until this point? I have my chat open and I have all of people's pictures up. So if at any point you have a question, you can hit the raise hand button, or you could put the question in chat, because I'll see it either way. And I did just want to highlight there are bigger mutations. So the one I showed you on the previous slide was kind of like in that sequence that we're familiar with in terms of like the DNA, we represent it as like a series of letters like ATCG. I was showing you small changes before, where it was just like, oh, just change a letter or add in a letter or delete a letter. But what we do see, occurring by accident in our DNA is bigger changes, where there's sort of like a swath of DNA that gets changed in some way. So what could happen is you could have sort of an entire piece of your DNA get copy pasted. It can get flipped around. It can get deleted. It can move from one chromosome to another. All of these are possible changes. I just wanted to show you here what that kind of looks like. This is a karyotype, which is when we take the DNA in a cell, we stain each of the chromosomes so that they can be kind of easily identified, and then we lay them out in size order. And normally people are supposed to have 2 of each one, and the 2 they have should look fairly identical at this level. And what you can see is that there's examples here where there's chromosomes missing. There's chromosomes that kind of look like they're a mishmash of 2 chromosomes because they have 2 different colors. These are the types of things we might see in cancer, where we just lose chromosomes, gain pieces of chromosomes, or chromosomes kind of get pieced together. We see this in cancer, and in fact, there are some famous examples of basically 2 chromosomes sort of swapping DNA, and that results at that interface where the swap happens in this sort of Franken gene that's like parts of 2 genes. So that example I gave of part cookie, part croissant. There's an example in cancer, especially childhood cancer, where there's a Franken gene that's part one protein and part another protein, and that actually drives a majority of a specific type of childhood cancer, just from that weird happenstance occurrence where these 2 genes come together, which is not typical. So these are all the kinds of mutations we might expect to see, but why do they matter? So if you're not familiar with how we talk about genes in the context of cancer, when we're usually looking at a mutation and we're like, okay, let's say that a gene was deleted. When we're looking at that gene and how it relates to cancer, we're thinking about things like, oh, is it kind of neutral? Like it didn't have any role in the development of the cancer? Or does it have a role, and is the role that when that gene was lost, that enabled cancer to happen because the cancer that was previously held back lost the thing that was kind of constraining it? Or is it a mutation where maybe something became overactive, and that overactivity actually allowed cancer to be able to do the thing that it wants to do? So we kind of think of it as, is it a gain of function or is it a loss of function in a gene that's relevant to cancer? And so if it's a loss of function in a gene, if that's relevant to the cancer, that gene might be what we call a tumor suppressor gene. So it's a gene whose normal function generally keeps things like cancer at bay. It usually holds things back. And so when you lose it, it's kind of akin to losing the brakes in your car. So the car just kind of doesn't know how to stop anymore. Versus gain of function happens in what we call an oncogene. And so these oncogenes are genes that normally do something that cancer likes. And so cancer instead, it likes that activity and then it takes it to 11. So it gains function. It becomes, let's say, uncontrollable. And we think that as analogous to when you're hitting the gas, but it's just jammed, like it just continuously is encouraging the car to accelerate. So those are kind of our paradigm for thinking about genes and their relevance to cancer. Is the gene more like a tumor suppressor gene, or is it more like an oncogene when we see it being mutated in cancer? So I just want to give you an example of how we get these brakes versus how we get these gas pedals. So an oncogene, I want to be very clear, is not a new piece of DNA, a new piece of gene that came from nowhere. There's actually a really interesting story in the history of cancer where we used to think that maybe cancers happen to us because of something external. So the way you could think of it is that we used to kind of think like, oh, there's like a virus or a bacteria or something that gets into us and makes us ourselves start acting abnormally. But one of the really cool stories was what we realized was that there are examples of cancers being caused by viruses, but what's actually happening is when that virus infects us, it actually is either giving us DNA that came from us originally, just it's a mutant version of our own DNA, or it does change our DNA, but just to make our own DNA more active, for example. And so we do have these genes that do promote things that cancer likes, right? Like we have our own cells that need to grow, our own cells that need to divide, right? Like that's how we develop from a fertilized egg into a person, is that our cells grow and they divide. So we already have that blueprint within us to do those types of things. It's more a matter of, in the case of cancer, you take that gene and then you turn it on when it's not supposed to be on, or it becomes way more overactive. Right, so just showing you here, it could be a slight tweak in the recipe, so then it's just super active, right? Like it's just way more potent than it was supposed to be, or maybe you have just a lot more of it, right? And so in the case of cancers caused by viruses, it was actually something that looked like this, where it was basically, we had that DNA already. It was just that the virus sort of came in and either gave us an extra copy or gave us a mutant copy or gave us sort of this hyperactivity. It gave us this way that our own genes got dialed up to 11 so that our cells would start acting cancerous. So it's just this kind of neat story in the history of cancer where we thought these viruses were giving us something completely foreign to our cells. And we actually discovered that what they were giving us was our own DNA, or activating our own DNA, and that it came from us originally. All right, so that's oncogenes. Again, this is something that gains functionality. It's something that cancer cells like, and they just dial it up. And then the reverse is the tumor suppressor gene. So as you can imagine, the way that our genes sort of operate in totality is like a circuit, right? So we have certain things that we will do, especially switching on in response to something about our environment, but it usually has these like feedback loops or self-regulation mechanisms, right? So often something will switch on, and then that, as a result of sort of the cascade of genes and how they interact with each other, it then switches itself off again, right? So there's a way in which a lot of how our genes have evolved is this circuit where things will switch on and then switch themselves off, just to prevent sort of, or runaway process. And so we have these things called tumor suppressor genes that are just inherent in our DNA. We like to have cells divide, but we want to do it in a controlled manner, right? We want our cells to divide, but we want them to just then stop and wait for the cue to then divide again. And so these tumor suppressor genes just normally exist in our body. And the idea is that when you have a mutation in the case of cancer, that protein gets deleted or it just doesn't function. There's something about it, the recipe that changed, and it just doesn't do its job properly anymore. And so it's just not holding that process back. And so one of the main examples of this that you may have heard of is a protein called P53. And so P53 has gotten this nickname as the guardian angel of the cell. And one of the reasons for that is it's just such a all-encompassing tumor suppressor gene. It's very sensitive to all of these different things that the cell is facing. So if the cell doesn't have enough nutrients, if the cell is in a low oxygen environment, if the cell experienced DNA damage, or let's say that it's in the middle of replicating and something went wrong, P53 responds to that. It senses that. And what it does is it mediates all these things that cancer doesn't like. All of these things do not serve a cancer cell well. So it either would cause the cell to die, it'd cause the cell to stop dividing, it'd cause the cell to enter a state where it never divides again. All of these things are sort of the antithesis of what cancer cells do. And so that's why P53 has sort of this central role in cancer. And in fact, mutations where we lose P53 and it doesn't work anymore are found in like 50% of cancers. Like cancers are very diverse in terms of what ends up getting mutated. It's not like there's one gene responsible for 100% of cancers, but the closest we come to that is something like P53. P53 is lost in most cancers. All right, are there any questions up until this point? Questions? So we talked about these different types of mutations, and I just want to be very clear that cancer is actually, as I said before, the accumulation of lots of these mutations. So it's not the case that you just get one mutation oncogene and then suddenly you would see the full-blown pathology of like stage 3 lung cancer. You usually need several mutations in a collection of oncogenes and tumor suppressor genes all together in the same cell for it to then start acting like a cancer cell and start developing into a tumor. And so the way you can think of it is this is sort of a timeline from left to right, where it's like, oh, starting from when we are a single cell, and then we start developing, and at that point we become a multicellular individual. We just accumulate mutations. And some of that actually is not even related to carcinogens. It's just a feature of every time our cells divide, they need to copy literally billions of letters, and they just make mistakes, right? It's kind of like if overnight you had to stay up and copy a 3 billion letter or character novel and just type it up by hand, you'll make a mistake here and there. And that's what happens in our own cells, is that they just make random mistakes as they're dividing. And so we're accumulating those mutations, and then we start accumulating mutations associated with our environment and our lifestyle. So that could be radiation, pollution, certain chemicals, and those start to accumulate. And at some point you might start to see then the onset of cancer. So if enough of those random mutations occur together in a single cell, that cell then might start, let's say dividing more than it's supposed to. And every time it divides, it's kind of like it's rolling the dice to see, oh, will I mutate again? And it's just by chance, if it mutates again, that could kind of keep it snowballing towards being a cancer cell. And so that's what we see here closer to the right, is that you start having like this like early tumor and early growth that's just more than it's supposed to. But at some point, as it keeps growing and growing, it could take on new abilities to be closer to cancer, which is not just about growth, it's about being invasive and really disrupting not only that part of the body, but also migrating to other parts of the body as well. And so that's what you see here more on the far right. So yeah, I want to just be very clear, it's a multi-step process. It's, again, just by chance are you getting all of these mutations all together in a single cell that lead to this cancerous ability. There's some lifestyle choices that do accelerate this, but again, it also is completely random as well. So when we're looking at what these mutations are perturbing, so we're mutating some genes, those genes normally had a job within the cell, and that job is being changed. Either that protein doesn't function anymore or that protein functions way too well. All of that contributes, all those different mutations, to the traits that we associate with cancer. And so if you haven't seen this diagram before, it's sort of like a seminal diagram in the field of cancer biology. We call this the cancer hallmarks. These are like the defining feature of cancer. And so some of these are a little more obvious. So things like the fact that these cells just keep on growing, keep on dividing. I think that's just what most people associate with cancer. But there's other things that are associated with cancer as well. So the fact that, for example, as the tumor starts growing, it becomes this big mass of cells, and it's a very hungry mass of cells. And at some point, normally cells are being fed by the blood vessels nearby. Those blood vessels bring oxygen and food. But when you have this giant growing mess of cells, like a developing tumor, they get super hungry and there's not enough food. And so they actually develop a way to recruit additional blood vessels to sort of bring them all the food that they need. And so that's an example of a trait that you might not associate with cancer, but it is actually very critical for cancer to advance sort of further and further along. So it's sort of all of these traits together are really what we see for cancer to get to a stage where it's like clinically observable and is something that needs to be treated. We have all of these things happening together. All right. Sorry, I just wanted to check something. All right. So I did mention this, but I do want to emphasize it again because it comes up a lot with how we think about the data that we get from characterizing cancer. And so we're going to talk later about, or we're going to talk in a second about how we characterize cancer. Cancer is sort of the result of all these genetic mutations, right? So it's the evidence is in the DNA, right? If you extract a cancer cell and look at its DNA, you can see all of these things happening, right? You can see genes being changed or lost or new copies sort of being pasted in. But I wanted to be clear that not every mutation you see in a cancer cell is directly contributing to the cancer being the way it is, right? We get lots of random mutations, and some of them don't really do anything, or maybe they do something, but it's so slight and it's not contributing to the cell being cancerous. And so the way we think of about them is what we call the difference between a driver mutation versus a passenger mutation. And so as we're growing, as our cells are dividing and we're accumulating these mutations, some of those mutations don't really have any purpose or any role. Like maybe they slightly alter a process in that single cell, but it's not enough to contribute to the cancer in any meaningful way. And so again, when I say you need the constellation of the right mutations all happening in the same cell, all those mutations need to be driver mutations. They need to actually be actively something that is part of the cancer process. They can't just be random mutations anywhere. A lot of those mutations either don't do anything or they just don't do anything that's relevant to cancer. And so you can imagine when we're sifting through this data where we have all of these cancer genomes and we're trying to figure out, okay, well, what went wrong here? There's actually going to be tons of mutations we see in the data that have nothing to do with the cancer. They're just mutations. They either do nothing or they just don't do something that's cancer relevant. And so that's something as sort of data detectives that we need to keep in mind, is figuring out we don't just want what is the most common mutation. We want a common mutation that is actually relevant to the cancer being the way it is. And I just bring that up to say that there's been a lot of efforts to characterize the genetics of cancer. So if you're not already familiar with TCGA, it's called the Cancer Genome Atlas. And as the name would imply, it's sort of this massively funded and coordinated effort to just collect many samples of a certain type of cancer. So for example, they might be like, okay, we're going to study kidney cancer. So they collect tons of kidney cancer samples from lots of different patients, and they sequence them all to sort of have this bank of data. Here is a genome example from kidney cancer patient 1 through 1,000. And the goal of this has just been to then try to figure out, okay, when we're looking at kidney cancer, what is being mutated? Does that give us more insight into how the cancer is happening or how we could possibly treat it? And you are welcome, like the data is in many cases open for study. If you want a more guided way to study it, I highly recommend cbioportal.org, which will let you search through this data set, but it actually guides you and does a lot of the visualizations for you. Chances are you could probably access the data on your own if you want more closer to the raw data, but you can start to see that cancers are actually quite different from each other. I'm not going to even fully touch on that in this talk, but if we're comparing a kidney cancer to skin cancer to brain cancer, there's actually quite a few differences in what we observe from the data from those different types of cancer. They all fall under the umbrella of cancer, but technically, depending on where the cancer is happening, the way it comes about can be very different. All right. I'm going to now transition into thinking about diagnosis. So that's sort of the background all about cancer, how it's caused, what is going on. And I hope some of that molecular context then helps make makes the data that we collect make sense. Because again, when we're trying to understand cancer, we're actually collecting a lot of data that's looking into what's happening at the protein level or at the DNA level or the mRNA level. So that's the data we're actually collecting and needing to sift through. So it just helps to see what does the mRNA mean? What does the protein mean in the context of cancer? Are there any questions before I move on to the next thing? All right. So let's start off. We're thinking about diagnosing cancer. So I'm just going to give you an example. So we have a neighbor. Our neighbor is Sasha. They are a local bartender. They like to hike. They like to play guitar. They haven't really had any serious medical issues. They do have a family history of melanoma, but otherwise, nothing that they've been diagnosed with in their own lifetime. But they start to experience symptoms. They start to get abdominal pain. They start to lose weight without the intention to lose weight. They feel nauseous. They oddly notice that there's pain in their leg. So the leg is kind of swollen, looks inflamed. There's a change in the color of their urine, and all of these worrying symptoms prompt them to go to the doctor. I'm just curious if you have any guesses. What do you think this could be? Anyone want to share in Zoom chat what this could be? Just had a random guess. Yeah, it's some type of cancer. So someone's pointing out maybe kidney cancer, given that there's a change in the color of their urine. Any other guesses? All right. So obviously, we're going to go to the doctor with all of these symptoms. And so the doctor is going to go through some sort of process to try to figure out what is going on. So I will say, when you're looking at cancer at the stage at which it's causing symptoms, at the stage at which a person is experiencing something like these different symptoms, at that stage, the cancer is usually large enough that you start to have what we call like gross features, like things you could just observe. And I just want to give you an example of that. When you're looking at a tumor, so let's say that you imagine you could kind of just like take a slice of the tissue if you know where to look. If you're looking at that tissue, you can already tell that something's wrong if it's at the stage where the person's experiencing symptoms. Chances are you'll notice the large abnormal growths, like the architecture of the tissue will be off, right? Like our through very interesting miracles that you can learn about in developmental biology, somehow our cells know to grow into certain 3D shapes, right? Like it develops into tissues and the tissues come together somehow in the correct way for us to be functional. And so when you're looking at cancer in a specific tissue, that architecture is way off. It's not just really necessarily that there'll just be this giant growth where there isn't supposed to be, but it often will sort of remodel a lot of the tissue around it, and you'll see things that are abnormal. I already gave the example of blood vessels will start to spring up where they don't normally show up because they're trying to feed this growing mass. All of that is sort of the influence of the cancer changing the layout around it. And so even if you just took a slice, you can kind of tell, hey, something's wrong here. And the joke was, when I was in my cancer biology research program, a lot of us was very molecular oriented. Not many of us had clinical experience, but even we could kind of look at these side-by-side histologies, these cutout tissues, and see, hey, this just looks chaotic. Like there's something wrong here. This is probably the cancer sample as opposed to the normal sample, just because of how messy it looks. And a lot of that messiness also is observable at the cellular level. So shown here on the right is a comparison of sort of normal cells versus cancer cells. And you can already tell that there's something way more chaotic about these cancer cells. They don't usually sort of sit nicely with other cells. Normally other cells will sit nicely in like a sheet and sort of all hold hands together. If it's a cancer cell, it doesn't hold hands. It's kind of more mobile. It's not connected to things. It's kind of going rogue. And it also just is very diverse in terms of like the size and the shape. Like normally they're fairly uniform if they're all cells in the same tissue serving the same purpose, but cancer cells don't obey those rules anymore. So they start to change in their size and their shape, and they lose a lot of the features we'd expect. And the one thing I want to point out is that they have these larger nuclei, and some of that is actually because of the fact that they might be duplicating chromosomes, and their DNA is like quite abnormal compared to the normal cell. So the DNA will look at, or the nucleus will actually look larger than we would expect. So we can see all these features if we know where to look. I'll just say that, obviously we don't necessarily know where to look. And so when we're trying to diagnose a cancer, we often rely on some sort of imaging. So we can use things like ultrasound, PET scans, MRIs to look again for something about the expected architecture of the area that looks different. Depending on the type of cancer, something that we think might be in the blood, we can look at a blood smear and just look at the cells that are floating around in our blood. If it's a skin cancer, because I know some people brought up melanoma, you could look across the skin. It's just like a physical examination, look for moles and try to diagnose based on the mole. There's actually certain characteristics of mole that tell them if it's a more dangerous mole versus a benign mole. All of these are sort of the ways that we would hunt around to figure out if we see something that's suspicious. Chances are, if we find something that looks suspicious, the best way to then really truly know that it's cancer is to do a biopsy. And so very often what might happen, I'm just gonna give the example of lung cancer. That doesn't happen to be the case in this today's case study. But if they were suspicious that you had lung cancer, they might have found that out by doing this imaging. So MRI or ultrasound, they might see like a mass that's like abnormal, like the density of the tissue, where that tissue is located, it just might be weird. The PET scan actually shows them where there's an area of high metabolic activity, which is more likely with cancer. As you can imagine, something that's growing and dividing so much is churning through lots of energy. And so that growing tumor is actually hungry. It wants lots of food and sugar to feed itself to power dividing so much. And so if you're looking at a PET scan, you don't expect this random mass of metabolic activity, but that might be a sign that points to a tumor in that area. And so let's say they have all of that evidence so far. The best thing they could do then, if they suspect maybe you have a lung cancer, is to take a tissue sample. And they don't want to do that willy-nilly, right? Like taking a sample of your lungs is not ideal, especially if you're, let's say, an older patient. But that might be the best thing to do if they really, truly have enough evidence to suspect that you have lung cancer. Because that's the most definitive, right? To look at the tissue up close, to look at the cells in the tissue. That is the best way to diagnose a cancer currently. Are there any questions about that? All right. So
I have one question,
okay.
Melissa. So when you talk about this biopsy, taking the cells out of this potential tumor location, so what doctor will look into is the sequence of DNA or anything else?
Yeah, so the first thing they will check is the histology. So they will really truly just want to look at an up close sample of the cells, right? So in all of the other things, they just see like this blurry mass, and a well-trained doctor knows that that blurry mass might be off, but really the best way to discern cancer is by looking at like a cutout of the tissue where the cancer lies. So they can see the abnormality of those cells and what they're doing. A well-trained doctor can tell like they're dividing rapidly. Look how much they are like abnormal relative to what lung tissue should look like and how much they've disrupted the area. So that would be like the first pass, just to really be able to diagnose this is actually cancer. You are right that they will often do some sort of genetic characterization if that will guide the treatment, or if it might tell them there are some cancers where we do sort of stratify, like, oh, it's not just you have cancer that is blank positive or negative, where blank is the genetic marker. So they might do some early genetic characterizations if it makes sense for the kind of cancer they're looking at. So for lung cancer, there is a genetic marker that's commonly looked at. So they might do that early characterization. They probably don't do something like sequencing the whole thing. That is kind of something they just don't do.
Okay.
Yeah.
So another thing is recently DeepMind accomplished this huge breakthrough in protein folding,
Mhm.
right? So can protein folding help anything in this domain to diagnosis or treatment? I'm like, sorry about my lacking knowledge in
No,
biology.
no, no, that's an interesting question, because I think one thing that I didn't emphasize is that cancer is by and large a genetic thing. And so, for example, by and large, what is making the cells abnormal is that their recipe for certain genes has changed. But technically, there can be ways that cancer is, I don't know if it could account for 100% of the cause, but the things that can contribute cancer can be technically non-genetic. So what we've noticed is there can be things that are not at the genetic level that actually encourage cancer behavior. And so what you're talking about, where it's like, oh, well, maybe the recipe for the protein doesn't change, but the way the protein folds changes. That could technically encourage cancer if the way it folds, for example, makes it less functional or more functional, right? Because basically the whole of what a protein does is based on its shape. And if the shape changes, what it does changes. So yeah, that's an interesting point. I don't know of any really solid examples off the top of my head, but theoretically, it's very sound to say if there was some way in which a particular gene and the protein it makes, the recipe didn't change, but the way it folded kept changing, that could contribute to cancer. I don't know an example off the top of my head, but theoretically that does make sense, because ultimately the end product, if it somehow still ends up being different, like it stops working, for example, that could be contributing to cancer.
Okay, thank you.
Yeah. Awesome. So let's talk about this first example, which is our friend Sasha. They get an ultrasound, a CT scan, a PET scan. I'm just showing you the images. They don't mean a ton to me because I am not a clinician. I'm very much molecular computational biologist. But if you are a well-trained doctor, you will notice that there are shadows or masses or bright signals not where they're not supposed to be, or in a shape that they're not supposed to be. And so ultimately, the doctor actually diagnoses Sasha with pancreatic cancer. And so my question for you all is, this is your friend Sasha, they're your neighbor, you've known them for a while, you hear that they have pancreatic cancer. What is your first question when you hear this news? And you can just type in Zoom chat, what would be the first thing that comes to mind? So I'm seeing How far has it spread? Is it recoverable? Like, can you be treated? What stage is it? Can you get surgery for it? All of these are very great questions. Yeah, you might have tons of different reactions or questions. I will say probably the most common one does get at this, which is sort of what are your options? How bad is it? That tends to be our first reaction. And what that's hinting at when we think about that is actually what stage is your cancer? And so if you're not familiar with cancer staging, that just reflects really how far it's progressed. So an early stage, so stage 1, is something that tends to be smaller. It tends to be very localized. So for example, if it's a cancer of the lung, it just shows up on one lung in one place. That would be an early stage cancer. As we move towards stage 2, stage 3, stage 4, that means that we suspect that the cancer has maybe moved from one lung to the other lung. Maybe it's moved from that lung to somewhere else in the body. Basically, the farther it's spread, that's where we have later stages. Stage 1 through 4 is usually what we talk about sort of in more common interactions. Technically, at the doctor's level, we have like a more sophisticated system of looking at things. Like we wonder, like, what is the relative size of the tumor and how much has it invaded locally? And then we will examine lymph nodes, because that's a very common sort of transit opportunity for a cell. If a cell is leaving one organ to go to another organ, the lymph nodes are one of the major sort of highways that they take. So one thing they might do is they might look into the lymph nodes nearby and see if they see a crazy abnormal lung cancer cell in the lymph node, which really stands out because normally in the lymph nodes, you maybe have like types of immune cells. You don't expect to see a lung cell in the lymph node. So they might check to see if they observe that. And again, depending on the patient, if they notice, oh, this patient has, we diagnose them with lung cancer, but we also notice the suspicious mass appearing on their kidneys, that is evidence of a metastasis, which is that the cancer has spread really from one organ to another organ. And so these do kind of map, stage 1 through 4 kind of map to this TNM system, but this is a little bit of a more detailed system showing you how they kind of arrive at saying you're stage 3. They're kind of looking at all these different features. And I will just say, it's not just size and invasion. Sometimes they are looking at the cells themselves. So for example, this is more common in something like prostate cancer, which is that it's not just a matter of like, how big is the tumor? How far is it spread? They actually are looking at like the cells themselves to see like how different do they look from what we expect? Like, do they look like what we expect prostate tissue to look like? Or how different are they? Because usually less normal is evidence that it's actually worse. So normally the prostate tissue is supposed to form these sort of like small little gland structures. And as you can see on this spectrum, as it starts to not really even form that structure anymore, that's a sign that the cells are just more and more abnormal. And so when you're staging the cancer and you look and you see, oh, like maybe one patient's prostate cancer looks closer to the top and one prostate cancer sample looks closer to the bottom, the person whose sample looks closer to the bottom, you'd actually expect that their cancer maybe is more aggressive or is harder to treat. And so that might be additional information that impacts your staging decision. So when we're looking at Sasha's case, Sasha's tumor measures 2 inches across. There's a presence of cancer cells in the nearby liver. There's high metabolic activity in 3 nearby lymph nodes. And then if you recall Sasha's symptoms, they have leg pain, they have abnormal color in their urine. How would you stage Sasha's cancer based on this information? Yeah, it sounds fairly late stage. And so I just wanted to point out something like leg pain could be evidence that cells maybe are in lymph nodes nearby and causing them to swell. As we mentioned, like someone's thought, oh, maybe it's kidney cancer because it sounds like it's affecting the kidneys. Well, maybe it wasn't originally from the kidneys. It's those cells have moved to kidneys and are affecting their ability to function normally. So yeah, this would be fairly late stage cancer. And so I just wanted to walk you through this example to show you really like this is the process by which we are understanding someone's cancer currently. I mean, I'm not a clinician, but this is by and large sort of the most common way. It's probably the most accessible way when we don't have the means to do tons of fancy genotyping and surveillance of someone's body. But this is just how someone would find out they have cancer. It's usually they show some sort of symptom. In some cases, they might be screened. Screening is a whole nother conversation, but they might show symptoms. They go in, they get some imaging done. They might then get a biopsy if they see something suspicious. And then that's where they start to sort of put the pieces together and figure out what stage are you, and that guides treatment, generally speaking. So this is a great, I don't know if you guys usually do like 5-minute breaks. This would be a great stopping point if people want a bathroom break.
Hi, Marisa. Just 2 minutes before we get started, I'm just very curious about the current vaccine strategy for COVID-19, because you have these new mutations keep on coming out, similar to the cancer mutations. So kind of worried about it.
Yeah, I mean, I think, so ultimately, when it comes to these mutations, like I'm to be honest, like immunology isn't super my strong point, but I will say that in general, when we're thinking about immunity, the COVID virus, right, like will have a particular shape and architecture, and ultimately what you're just hoping is that your immune cells will realize this is something really problematic and have like a strong enough rejection response fast enough, right? You don't, like, for a lot of people, the issue is that they do have something somewhere that will respond to a COVID virus, but at that point, the COVID virus is starting to infect, the infection is spreading. And so it's kind of like they've been stealthily invaded before they mount a large enough response fast enough. And so with the vaccine, trying to get people to like really have that quick, immediate reaction right away to just reject it. I think there can be mutations in viruses that aren't necessarily changing, like, you know, it's like a tiger changes its spots. Like, it's some of those mutations aren't necessarily changing the outside arrangement that we recognize. I mean, some of them do. I think that's something that's really tough with, you know, things like the seasonal flu. It's like they are changing their spots often. And so we just don't have the same immune reaction quick enough as it starts to change the features that we recognize, right? Like the things we're looking for are literally changing. Some of the mutations might change things that aren't that particular protein on the surface. They could be other things, right? But yeah, I think as long as the tiger is changing its spots and those features are changing, then yeah, it could be that our immune response to them is weaker, and at some point maybe we don't have the same immune response at all, and then we need a new vaccine to like prompt us to respond to that one instead. So I think some people who've said, like, depending on the degree of the mutation and whether it's always kind of in these recognizable outer proteins, that might prompt us to just keep having like updated vaccines the same way we do the seasonal flu. But it could be the case that also is mutating in ways that don't affect that surface protein.
Okay, thank you so much.
Yeah, I notice we're at time, so I'm going to resume. And now we're going to sort of really segue into thinking about where does machine learning and AI come in. And so honestly, this is kind of, when I'm thinking about machine learning, this is kind of, again, how I break it down, right? Thinking about the difference between a supervised task versus an unsupervised task and all of the methods associated with those. And I just want to give you examples of the types of things we think about in cancer that relate to each of these areas. So for example, a lot of work we do is unsupervised. And I'll just say that that's because with something like biology, there's so much open-endedness where we're just trying to discover what is happening, what is this, how does it work? And so there's a lot of unsupervised tasks. So there might be something like figuring out what are the different types of tumors in patients, right? So we might have some big disease like all of lung cancer, but maybe if we're sort of doing an unsupervised analysis, we might realize, oh, there seems to be these different types of lung cancer all under this umbrella. And so that's just at the level of looking across patients, but you could go a level further and say, oh, well, we're taking samples of lung cancer cells, and you might realize, oh, there's different types of lung cancer cells, right? It's actually kind of a heterogeneous space. Not every lung cancer cell in one person's tumor is actually the same. And so as we're doing sort of this unguided analysis where we're just looking for what is there, what is the diversity there, are there certain clusters or archetypes, we might discover, oh, in lung cancer, we tend to notice that there's this common cell type, alpha, beta, just I'm making something up, but like we do a lot of this type of analysis just because previously we haven't had this level of detail available to us about lung cancer, either at the single cell level or just not enough samples to be able to appreciate what is there. And so we do a lot of unsupervised work, a lot of visualizations, a lot of clustering to just figure out what is the diversity of things within a given cancer. We do a lot of supervised tasks. So we do a lot of trying to be able to better discern normal patients versus tumor patients, or trying to relate tumor types to like their prognosis and their survival. So we do a lot of these more guided tasks. They tend to be the much more clinical ones. I'd say the unsupervised ones tend to be the more figuring out how cancer works. We're still many decades later figuring out how cancer works. And a lot of the supervised ones tend to be very clinically relevant. Like how can we automate sort of this ability to diagnose patients just based on a certain type of data that we collect. And so there's lots of things that relate, I think, going from machine learning into cancer biology. And we have lots of data. I think the tough thing with biology is our data is very messy. As soon as you work with real world, real life things get very messy. But I will share that we measure many things in biology. We measure many different types of things at different levels of scale, at different levels in the biological process. We have things like sequencing, we have things like imaging, we have things where we're looking at the spatial arrangement of cells. We might be looking at the proteins in a given cell. So we have all this kinds of different data that we look at. And I'm going to go through and explain a couple of these different data types and how they relate to cancer. So we talked about this already, this idea that we have the DNA, which is our sort of recipe book. It tells us, how do you make any given protein? And so the DNA and what's going on in our cells is the DNA is being copied into RNA, which is sort of this temporary intermediate. It's kind of like a photocopy of the recipe book. That photocopy floats out and goes to a ribosome, which is where the protein is constructed. So it references your photocopied recipe and uses that to then put the right pieces together to make a particular type of protein. And so if we're looking at these steps, we basically can collect data at each step of this process. So we can collect data about the DNA. We can collect data about the RNA that's floating around in our cells. We can collect data about what proteins we have floating around in our cells. I will just say one underappreciated part of this process is actually that our DNA isn't just sort of this like, it could be like a text corpus, right? Like it's a lot of letters in a certain order, but it's also a 3D physical thing, right? Our DNA is these molecules, and these molecules actually have a lot of folding and sort of coiling that's happening. So there's this really cool world where they look at like the 3D structure of DNA and how DNA basically turns into like knots and coils. But I just bring up this up to say that actually that 3D shape matters in the sense that when we're expressing genes, if a gene is sort of super coiled, it actually becomes sort of hidden away, like it's so folded up that we can't reference it anymore, and that gene isn't expressed. Versus if there's a part of the DNA that unfurls and uncoils and is very accessible, that gene is easier for the cell to express. And that that shape that it takes on is actually very purposeful. In certain cells, they will fold up parts of their DNA that they're like, I don't need to use this anymore. And they'll unfurl the parts they do need to use. And that's usually relevant to the type of cell it is, right? Your DNA has the instructions to be any type of cell in the world. But if you're a lung cell, you don't need the instructions for how to be a kidney cell or how to be a skin cell. So you fold up that DNA and you reference the parts that are relevant to being a lung cell. And so that 3D architecture actually really matters. And I will talk about an example of data where we look at what genes are more open and more accessible versus which genes are closed. And that is relevant to looking at cancer. And then one other thing I'll mention is that our notion of cells is that, this is actually kind of relevant when we were talking about the viruses, is that the surface features of a cell usually tell us a lot about the cell, like what it's doing and what kind of cell it is. And so even if we weren't diving in and looking at, hey, what is the cell sort of thinking and doing by studying its DNA or studying its proteins, if we just look at the outside of the cell, there's usually like flags that tell us what the cell is, like what kind of cell it is. And so a common example is that if we just look at your blood, if we take a sample of your blood and measure certain flags that are present on the outside of the cell, we can say, oh, this is a white blood cell. Specifically, it's a T cell, or this is an like an innate immunity cell. It's a monocyte versus a red blood cell. Like those are things we can detect looking at the flags on the outside of the cell. So these are all things you can measure. We can measure what is the DNA, what mRNA is it producing, what proteins is it producing, what proteins are on the outside of the cell that make it a certain type of cell versus a different type of cell. And we can also tell not only like the DNA in terms of what mutations it might have, we can also tell what kind of shape the DNA is taking on. So these are all different types of data that we collect. Are there any questions before I kind of get into those? All right. So probably the most obvious one I just talked about, TCGA. We do a lot of DNA sequencing. This has been aided by the next generation sequencing. We used to do sequencing in a very labor-intensive and cost-intensive way. And now we have new technologies where we can do DNA sequencing a lot faster and a lot cheaper. And so this is a commonly cited plot where before it used to cost, back in the 2000 era, it would cost millions of dollars to just sequence a single genome, right? It was a huge effort, took many labs, millions of dollars to get one genome. And nowadays, maybe in a week or two, you could get a genome for closer to like $1,000. And it can get cheaper and cheaper all the time as the technology gets better. So that has allowed us in many ways, whereas before we couldn't study the genome of a cancer, now we are able to collect those samples and actually look at their entire genome as opposed to just looking at one mutation at a time, for example. I mentioned the example of looking at the shape of the DNA. So like, is the DNA very open or is it very closed? There's two ways we detect this. So there's this thing called DNA methylation. The long and short of it is that with DNA methylation, the DNA gets like a chemical flag, like a little modification. And that modification basically tells that DNA to shut down. The DNA doesn't disappear, right? Like the cells don't usually want to ever get rid of parts of their DNA, but they do like to sort of tag something and say, okay, we're done with this gene. We're not going to express it anymore. And so methylation in this case is just this little flag that says, okay, we're done with this gene, close up shop. We're going to use something else. And we're able to detect that using a particular type of sequencing. So it's not just sequencing the DNA, you're sequencing the DNA to look for where that modification shows up. And so I could tell you, hey, it's not just what the recipe reads, it's that, oh, this part of the book has been sort of post-it note, like this is the actively referred to part of the book, versus parts of the book that are almost like taped shut, like they're just done with, the cell is not using that DNA anymore. And so this gives us a better picture of like what the cell is in the midst of expressing at any given time. So there's that methylation flag. Another thing that maybe is a little more physically clear is that we can actually look at the folding of our DNA. So normally our DNA gets kind of coiled in a way where it's kind of like beads on a string, or like if you're winding up a hose, there's kind of like a spool and the DNA goes around it. But some parts of your DNA will have sort of like DNA wound around spools and then wound around each other, and other parts will have the DNA kind of loosely associated with the beads, but otherwise it's very open and accessible. And so that's shown here on the left, where there's the closed chromatin, the closed DNA, very tightly wound. You can't really see what's happening in there. And there's the open parts of the DNA. And so recently, and this was actually at Stanford, they developed a way to specifically figure out where are these open regions. And so they developed this clever technique where something comes in and tags the open regions because, well, the closed regions aren't accessible to that particular enzyme. So enzyme goes in, it tags all of the open regions of DNA, it can't get into the closed regions. And then there's a way at the sequencing level to see, oh, this is the open region where there were lots of tags. And when there's no tags, oh, that was the region in that DNA that was closed off in that particular cell, just based on how it coiled. So this isn't even a chemical flag anymore. It's just based on like, oh, this part of the DNA kind of folded in on itself. And so it's not being used. So this is another piece of data that is also available and becoming more available all the time. So let's look at mRNA. So we've moved from DNA, how it folds, what mutations are present. Now we're moving to the next level, which is we're photocopying the DNA to get RNA. And that tells us really what, this is getting closer to what the cell is actually doing, right? So the DNA, with all those mutations, they only matter if they affect what the cell creates, right? The cell might have certain mutations, but if it's not producing the mRNA and protein, then the mutation kind of doesn't make a difference. It's just sort of what the cell is, but not what the cell is doing. And so when we look at things like mRNA and protein, we get a better idea of what the cell is actually doing, because the mRNA and the protein are what the parts of the cell that do things, as opposed to DNA, which just sort of sits there. So how do we characterize mRNA? There's a way in the past that's very quick to just characterize the relative amounts of different mRNA. And that's what's being shown here with these microarrays, which is where they basically take they chop up the mRNA, they adhere it to probes on a chip. And basically, if there's less of the probe or more of a probe, it's like a relative measure, then it turns different colors. So if you compare two samples, let's say a normal lung tissue versus a lung cancer tissue, the relative proportions of each type of gene will stain different colors. So if there's more in the lung cancer sample, it might turn red, versus if there's more in the normal sample, it might turn green. And so you get this relative measure of how much mRNA there is in, let's say, tumor versus normal. And this was the original way we did it. It was cheap, it was fast, and just gave us this readout on genes that are maybe expressed more in cancer or expressed less in cancer. What we actually use now, which we were talking about a bit, is RNA sequencing. So it's kind of the same as DNA sequencing, but it's at the RNA level. So rather than collecting all of the DNA and all of the genes, we collect all of the RNA that the cell has happened to create at that given time. And we figure out, okay, well, what genes are represented in these mRNA? What is it in the process of making? And so it works very much the same way in terms of like the reading out the sequence. But the nice thing about it is it's both quantitative and descriptive. So RNA sequencing can tell you the relative amounts. Like if there's a lot of a certain gene, you know, like, oh, there's like, this is a rough, like there's like on the order of a lot, like 20, 30, hundreds of this one mRNA transcript. It's expressing a lot of this gene versus very little of a different gene. So you do get these quantitative readouts, but you also get descriptive readouts. So even if you hadn't sequenced the DNA, you can tell at this level if, hey, we know what gene X is supposed to look like. We can tell that this mRNA is off. It's been mutated in some way because the DNA it's referencing has also been mutated. And so at this level, you can both characterize mutant instructions for making proteins, but you can also quantify how much mRNA is being produced. Like, is it telling the cell to make a lot of a certain oncogene, for example? And then sort of the last in this progression, so we talked about DNA, RNA, and now we're talking about protein. There are ways to quantify protein. It is harder, I would say, than DNA and RNA. DNA and RNA are nice in the sense that they are these like little linear molecules, and they only have like 4 distinct options. They either read, ATCG if you're DNA, or AUCG if you're an RNA. And just knowing that they take on a certain shape and that they only have so many letters at a time makes it very easy to read and reference them and measure them. Protein is actually a lot harder, right? Protein is like this physical 3D glob that takes on different shapes. And so when we're trying to measure protein, it's harder. We can use things like mass spec, where we try to look at the chemical composition, basically, of a protein. Or we can do things that are antibody-based or probe-based. So we could, for example, engineer to like look at a particular protein that has a little light on it, basically. So it's like, oh, it lights up green versus it lights up blue. Or we could add antibodies, which recognize the protein, and they are basically the flag that says, oh, I added this antibody. The antibody has a little red dot on it, and it looks for protein X. And so by proxy of how many antibody I see binding, that tells me how many of protein X I have. And so these are all very indirect measures. So measuring proteins tends to be a lot more difficult, unfortunately. But in some ways, it's very impactful. As I said, this will tell us, it could tell us what kinds of cells we have, and it really does tell us directly what a cell is doing, because proteins are what the action comes from in a cell. So it's like one of the most important end stages of the process, but it's also the hardest to quantify. Are there any questions about that? All right. I just want to touch on 2 things, which is that when we're looking at cancer, there's lots of levels. I think that's one of the hardest things for a lot of students in cancer biology to sort of wrap their mind around, is that we move through these different levels of scale really interchangeably, right? There's like populations of people who get cancer. There are, cohorts who get certain types of cancers and have certain lifestyles. Then you zoom in on a patient and what the patient does. Then you zoom in on their tumor, but their tumor is actually a collection of very different cells. So then you zoom in on a particular type of cell, you zoom in on the DNA, and we're like moving through all of these levels to talk about cancer as a whole, which I think is the hard thing to navigate. So everything I've talked about up until this point was sort of looking on a molecular level, either at one particular cell or like all of the cells like mushed together into like an average. But technically, all of the cells are special, right? Like if we're looking at a tumor, the cells actually can be quite different from each other in terms of what their mutations have ended up being relative to each other, and also what they're doing at a given time, right? So this picture here is just showing you like a tissue is like all of these cells collected together. And actually, when we look at a tumor, it is incredibly heterogeneous, right? There's lots of different types of cancer cells. There's actually normal cells kind of interwoven in. They've either been recruited in, or they're just sort of like bystanders in this process. And so you can think of it as like when we're trying to take a sample and do DNA sequencing, we're actually just grabbing a whole bunch of these and just mashing them together to do the DNA sequencing. And so that adds to the noise. It also just adds to the importance of trying to discern spatially what's happening, right? Because these are actually all different. Like there's an architecture to all of these things interacting, and we just sort of fuzz that over by like taking a giant scoop of them and mashing them up and measuring them. And so I just want to mention there is a lot of effort to try to look at them in their original site, like where do they sit relative to each other? And this has been done for a long time. So I talked a bit about histology, where you just take a slice of a tissue and you look at it. But in the past, it's been very hard for them to look at more than 1 or 2 things at a time. Like they see the physical layout and they maybe could look at 1 or 2 proteins at a time. But there's been a lot of effort to try to look at multiple things like proteins or RNAs or types of DNA in their current layout. Like, so where do cells that are doing certain things sit relative to each other within a tumor? And so that's an active area of interest, is trying to be able to do those measurements, but without extracting the cells and like mushing them all together, because that just sort of fuzzes away a lot of the complexities of the system. All right. Any questions? All right, so let's see. Let's go a little ahead. I wanted to give an example of a couple of these applications. So I just want to say lots of different types of data that we're collecting. Some of these I haven't even talked about. Trying to integrate them is really challenging. Like there's such different types of data measuring, things that are related but different. And there's a lot of people interested in putting that information together, right, to have a more complete picture. But that is a very challenging thing to do. I will say that this obviously fuels diagnoses, like better understanding what kinds of cancer are out there and which kind of cancer you have. But obviously, it also guides how we treat the cancer, which we'll talk about next week. All right, so let's talk about examples in the world of diagnosis that have to do with machine learning. So let's talk about discovering different types of cancer cells. So I just want to mention that we do a lot of clustering based on markers in the sample. So maybe we collect a lot of different DNA profiles, and we've done a lot of different work clustering to say, oh, like it's not just lung cancer, it's lung cancer type A, B, C based on their genetic profiles. So we've done a lot of work in that area to sort of discern, like it's not just one homogeneous group, there's lots of different types of lung cancer. Can we sort of de novo discover what those types are on the basis of their genetic differences or protein level differences? But I just want to remind you that clustering could be performed on many levels. You could cluster patients, you could cluster tumors within patients. Some patients have multiple tumor samples. If a patient has a metastasized cancer, you could literally collect like 3 or 4 different tumor samples just from one patient, like one from the left lung, one from the right lung, one from the kidney, one from a lymph node. And so are you clustering the patient, or are you clustering the tumors within a patient, which is different? Or you could cluster the cells themselves, which is an even smaller level of resolution. So we've done a ton of that, and you can actually find in the literature that we've discerned different subtypes of breast cancer, lung cancer. This has been done in many different big cancer types, where we've realized there's actually a lot of diversity of subtypes within that type. I just want to give you an example of discerning normal versus tumor. And so I just want to say that in general, doctors are very well trained to be able to see when something is off, right? They can look at an image and say, hey, this mass is not what I see in all the 50,000 other lung images I've looked at, like this is abnormal. And so doctors are experts, they're usually very good at that, but there are definitely cases where experts differ, right? Get a second opinion. 2 experts who've seen different samples over their lifetime and are maybe sensitive to different features in the image might say different things. And so this is where maybe machine learning could come in and either sort of standardize the process, maybe see something that humans miss. But this is an opportunity, I think, certainly where we could bring in machine learning and just help this process. Like, can we sort of learn from many, many experts and sort of pile all of their knowledge together to have a more accurate way of just looking at images and saying, hey, this is probably cancerous, you should follow the biopsy, versus it's probably nothing, don't worry about it. Just because, again, humans are biased, humans make mistakes. Could machine learning do it better? And then here is a slightly longer example that I just want to give. This actually this kind of work comes out of my previous PhD lab. It wasn't my work, but it was work done by a colleague of mine, which is trying to, again, look at sort of the difference between normal versus tumor in patients and helping with the diagnosis, but also relating those different types of tumors to how well they're likely to do. So a lot of profiling these different patients, again, is trying to figure out like what subtype of cancer you have. You have lung cancer, we discover it, you have the symptoms, we diagnose you with lung cancer just based on the imaging and maybe a biopsy. But if we measure flags, like genetic flags or protein flags, A, B, C, can we group you into type A versus type B? And do those types do better or worse? Maybe you have a more aggressive cancer, and that guides us to then be more aggressive in how we treat, versus something that we can maybe be a little more like wait and watch and see what happens. So there's a lot of interest in doing that. I want to say, I gave the example of, is this lung cancer? No, this is not lung cancer. I apologize. This is just showing you that we're developing our understanding of these different genetic markers and which ones are relevant in a given cancer. And so this is just showing you that, back in 1987, we realized that there was this protein that is commonly mutated called KRAS. And so we would just sort of stratify patients into, do you have a KRAS mutation or do you not? But then over time, as we've discovered more genetic markers, then we can sort of create more buckets and have a little bit more guided therapy, right? So then we discovered EGFR. And so the way we treat an EGFR patient versus a KRAS patient within lung cancer might be different. And now we have way more buckets. As we've gotten way more data, you can see on the right, we have so many different markers we can look for that guide how we diagnose and then how we treat. So here's the example from my lab, which was looking at pediatric patients who have a type of cancer that occurs in the blood. And so this type of cancer, you can take a bone marrow sample because bone marrow is where blood originates. So a lot of the different blood types come out of precursor cells in the bone marrow. And so you could take a sample of that bone marrow and basically see what are the kinds of cells that are in a patient's bone marrow, especially if they have cancer. And what this researcher did is they basically were looking at samples. They had ability to kind of look at the different types of cells in different patients' bone marrow. So you can imagine they took maybe my bone marrow and Ed's bone marrow, and they can basically discern oh, how much of each type of cell do you have? And do these cells look normal or do these cells look abnormal? And so they basically created the spectrum of how much of each type of cell do you have? Does it look more abnormal? Does it look more normal? And what they did is they related that, the amount of each type of cell and how normal it looks or how abnormal it looks. They related that to whether that patient would ultimately relapse or not. So they basically know of their sample, how many patients had a more aggressive cancer, had a cancer that was ultimately resistant to therapy versus patients who responded well. And they wanted to figure out what is special about those patients who tended to relapse. So if we know that ahead of time, we can be more aggressive in how we treat their cancer. We know that we need to be careful with that patient because they're very likely to not do as well. And so they basically went in and tried to learn the features that distinguished patients who relapse from patients who do not relapse. And they were basically able to figure out, hey, there's sort of this signature of things happening in certain cells that is unique to the patients who tend to relapse. And that could actually guide them to think about, okay, that profile, those things that are different about the patients who relapse, can we target those ahead of time so that they're less likely to relapse and basically preempt what we've observed will tend to happen? So I'm happy, I just realized that the reference to this paper is not in my slides. I'm happy to add that in for next time, just so that you're able to look up this paper if that interests you. All right, in the few minutes I have left, I just want to call attention to the challenges in this area. So I've highlighted some more success stories, but what is so hard about machine learning in biology and in cancer specifically? So I want to say a couple of the problems are very technical and a couple of the problems are not very technical. So the number one problem I'll actually bring up is not technical at all. It actually has to do with crossing disciplines. It's been very challenging when there's people who's been traditionally trained in computer science and algorithms, and then people who've been trained in the wet lab biology and they know how these measurements work. Having them talk together and design a study that is rigorous and sound has been challenging because they don't understand the limitations on both sides. I've dealt with people who don't understand how RNA sequencing works. And so when I tell them, hey, it's this average of all this mush, they're surprised. They're like, oh, can't you get out who contributed what? And there are some ways that you can kind of do that, but fundamentally you're just mushing the cells all together. Method wise, it's an average. And that's been something they weren't aware of. And other people who don't understand the algorithms might assume that you can back solve things that you can't. So that's one inherent problem in this field, is just the siloed off disciplines and the lack of communication. But there are a lot of other issues. I mentioned noisy data. huge variability within the data. Like you can have 2 lung cancer patients who actually, despite both having lung cancer, seem incredibly different from each other in their pathology and what their tumors say. I did mention the idea of integrating multiple types of data. It's very hard to bring together the mutational profile and what the DNA says and what the protein says and make them sort of work together to have one picture. I will also say that it's very tough these days. I think you might have heard of this idea of personalized medicine, but if you think about it in terms of how you would go about setting up a machine learning problem, the more sample, the better. And so when we're emphasizing, hey, personalized medicine, have an N of 1, that starts to fly in the face of the statistical rigor we need to back up what we're finding. So I think I will leave it at that. I have these slides. I think Ed, you might have these slides already uploaded. If not, I'll send you, this is a slightly updated set. I mostly actually took away things rather than adding things. I'll send the updated set, but I think I will leave it off there. And I'll just say that next week, we're going to really focus on, okay, we can figure out these different cancer subtypes, but how do we discern how to treat these different patients and use AI to do so better?
Okay. Thank you so much, Melissa.
Yeah,
Questions from students?
we'll have plenty of time also next time for questions, I think.
Yeah, so you can leave your question on Slack. So next time when Melissa comes back, and she can answer your questions.
Awesome. Thank you so much, everyone.
Yeah, so much. Thank you so much. Looking forward to seeing you again. And remind the students, your proposal is due, the project proposal is due tonight. If you need more time, just let us know. And next Tuesday, I look forward to your presentation on your proposals. And Sahil, please try to organize the discussion next Tuesday. And I will come back to see Melissa again on Thursday. Yeah. Okay, thank you. And
awesome.
bye, everyone.
Thank you, everyone. Bye.
Thank you.
Thank you. See you next week.
Okay, thank you. Yeah.
Yeah, thank you so much. I will see you next Thursday. I think the slides I have sent you in the past